Okay, today we're here with Dr. Joshua Doloff. He is coming to us from the Department of Biomedical and Material Science and Engineering at Johns Hopkins University. And title of his talk is quite interesting: Improvement of Islet Engrafts via Treg Induction using Immunomodulating Polymeric Tolerogenic Microparticles. Big mouthful there, but actually he's spoken with us before, and his work has really been progressing at sort of the speed of light, I'd say. So I'm very excited to have him here. I don't know if you want to say a few words just about your background, Josh.
Yeah, I'm actually going to give a little bit of an intro of that as I go through the initial slides, just to talk about where the field was and is and moving towards for type 1 diabetes and cell graft transplantation. But yeah, I did my PhD at Boston University, did my postdoc just across the river at MIT, and been at Hopkins. It'll be my 5th year anniversary, November 1st this year. So I'm excited to be here and to talk with everyone.
Fantastic. We have a nice international audience, and I'm going to just give a shout out to say that if you have any questions or comments, please drop them into the chat. And then at the end, we'll have some time for Q&A. Thanks so much for joining us, and take it away.
Sure. So our lab tends to focus at the interface of immune modulation, specifically any disease state that where the immune system is essentially involved. And this historically has involved implant as well as transplant rejection response. Our tack has been to really sort of just using systems biology tool sets, not just like a correlative kinetic responses over time, but to actually knock things out, to probe further, to establish causality, to know when something is removed, you see an appreciable change in the system, which means that it's a legitimate drug target. It's important in the role and in the response that you see. And so as a consequence of that, I'll be talking a little bit about some of the technologies today that we've done historically regarding drug delivery and drug formulation, and as well as also getting into a little bit of today's topic from a recently published paper in collaboration with Jordan Green's lab at Johns Hopkins called The Improvement of Islet Engrafts via Treg Induction using Immunomodulatory Polymeric Tolerogenic Microparticles. And so we tend to call them TOL APCs for tolerogenic antigen presenting cells, if you will, too. So that's another concept that's come out of the lab. Sorry, I just saw your chat message. Okay, so our tack can, the way that we approach diagnosing host response, it can really be done at the macro scale level, and it can also be utilized and put to good use down to the microparticle or even nanoscale level too. And so you can see historically immune cells such as macrophages or other antigen presenting cells can phagocytose, clear particles through the circulatory system or through excretion. I'm going to give you a little bit of background first, though, in terms of more macroscopic devices that are considered foreign bodies. So when an object exceeds the size of about a single cell, around 5 to 10 microns, it actually goes through a process of what we call frustrated phagocytosis. And instead, from the left-hand side, you go over to the right-hand side, where you actually elicit a very prominent immunologic attack and sequestration, sort of scarring over of a device. And this is actually a hydrogel capsule encapsulating primary islets for type 1 diabetes transplantation. So in my earlier postdoc work, I really focused on breaking apart what were all these immune cells, which ones were absolutely crucial and important to the response. And with one of my colleagues, who's now actually a professor at the Technion in Haifa, I'll show a photo of him with me later in the talk, but he and I developed crystals for preventing fibrosis, co-encapsulated with islets, and this went over, drug release went over 1.3 years in rodents and over 6+ months in non-human non-human primates. So very exciting work. So just to reorient you here again, whether it is biomaterial alone or having a biologic graft inside, what we now appreciate actually, and I'll show a little bit of data on this, is that biomaterial response is a very unique foreign body rejection response. When you put a biologic inside, it also makes things complicated, because from that graft, you also get secretion of antigens. And what we realize for type 1 diabetes, believe it or not, is a lot of the autoantigens that cause autoantibody formation to become pre-diabetic usually takes 1 or 2 autoantibodies to be pre-diabetic. And by the time you have a 3rd or even a 4th autoantibody to different antigens, you are considered full-blown diabetic. And believe it or not, a lot of the antigens that have been identified that are implicated in that progression are actually secreted molecules. So there's a lot of really cool work in the field being done with MHC modifications, hypoimmune cells, if you will, where you don't even need to have an encapsulating biomaterial anymore. But we have suspicion to believe that it's complicated. It's more complicated based also on the secretome of the cells. And so even though we can block foreign body response fibrosis, we do appreciate that this other indirect secretion of molecules, the immune system being present outside of the capsule, can sense them. And believe it or not, Sherry Stabler's lab at University of Florida showed a very elegant in vitro study. We've talked about this for years, but her lab actually showed it definitively, that the immune system can actually secrete cytotoxic factors back in through the hydrogel and stress and even kill the graft inside indirectly. It's very powerful data. But to show you really typically what happens when you don't have a graft, so in a biomaterial or an immunoisolation device, usually there's cell contact requirements for a cytotoxic lymphocyte, whether this is a natural killer cell or a T cell, CD8 T cell, to butt up to a target graft cell. Perforin is a protein that allows for the perforation and influx of many cytotoxic granzymes. You can see within the first hour of contact, the recipient cell, in this case a tumor cell, the shape changes, and by the second hour, these granzymes go downstream to cleave many substrates throughout the cell, including mitochondria, spiking creating intrinsic apoptosis induction. They even cleave many targets in the nuclear lamin structure as well. And you can see the cell within even just 2 hours starts to bleb and fall apart. All right, so again, foreign body response really is an outside attack. I was working with Bob Langer in my postdoc and Dan Anderson, and I asked Bob a very naive question. I said, is this some sort of plastic sheath you put around this device? It's a microchip device about the size of a USB key, receives wireless signals, it induces an electrical impulse. A gold nanowell cover is melted away and drug contained within is released. And this was actually this came out of a human patient over 6 months for an anti-osteoporotic study and anti-osteoporosis. And you can actually see this kind of very smooth, thin overgrowth. And when Bob showed me, no, that actually is the foreign body response, he says very globular type appearance. When you look histologically, you see very thick capsule surrounding the outside of the device, which is by Mason's trichrome in the bottom right, very heavily laden with blue collagen. All right, so and then historically, we've been able to identify that it's not just proteinaceous overgrowth. There are actually vibrant cells based on DAPI nuclear staining. You can see many cells on the surface of these capsules. Historically, it was a really great kind of testing ground, though, for us to be able to start parsing apart this pathway, because you can see whether these very translucent hydrogels are partially fibrosed, more completely fibrosed, or even where that fibrotic plaque emanates outward and connects material to material or material to tissue. And so there's sort of different gradation states of this phenomena. And so I came in trying to understand immunologically. There was no one on the team yet focusing on the immune response, and really trying to identify unique markers to identify a response. And so this was done by manual target by target qPCR validation, because even though there were, for instance, pre-built inflammation and nanostring probes as an example, I think we tested 179 probe set that they had originally years ago, and only 10 or 15 things lit up. And we were like, well, does this mean that the material is biocompatible? It doesn't make sense. We know that the material gets worse over time. It was based on my conversation with NanoString. I went back to them and I said, where did you get these targets? And they said, oh, from our collaborators in academia. And I said, well, what are they working on? And they said, cancer. So they had compiled all these, they had basically compiled a biased probe set that was not as relevant for a model system. So always be aware of the tool sets that you guys are using, because it can definitely affect your impression of a situation. So with the 3 bars that you're seeing here, the first one is non-implant. The second one is a mock implant procedure where we do the surgery, but we just inject saline or PBS. The third one is where we have hydrogel alginate encapsulating biomaterial present. And you can see macrophage markers, F480 CD68 spike following 2 weeks of implantation. C11b is a general myeloid marker. LY6G is a neutrophil marker. CD19 is interesting, is a B cell marker. We weren't expecting that. And then alpha small cell actin is now well-known fibrosis marker for activated myofibroblasts. And there are many collagens. Collagen 1A1, 1A2 are typically mostly shown in a lot of studies, but there's a very dynamic collagen isoform landscape. And then many of these other immune populations, interestingly, dendritic cells, which we have CD11C now included in 2 of our new data sets, and KP46 for innate natural killer cells, CD4, CD8, and FOXP3 T cells, and FOXP3 for Tregs. I grade these out in the original paper. The original paper was actually focusing on just biomaterial response alone, but when you put a graft inside, we know that these other populations, which are heavily involved in antigen presentation, as well as also targeted attack based on that perforin granzyme pathway, natural killer cells and CD8 T cells come in to try to kill the graft. And many of these populations engage in response.
Josh, while we're on that slide, can you just unpack why you were not expecting the B cell and like what that might mean?
Sure. So, really, I looked into this quite extensively. B cells, when I saw them responding, they were delayed by about 7 days, which made me think, oh, well, they're an adaptive immune cell, so surely they take time to respond and they're just engaging later. But what was very interesting about them is when I looked systemically in the blood of these animals, there was no IgG or IgM or antibody in general secretion. What was interesting is the B cell receptor, the IgM moiety, was being retained on the surface as a B cell receptor, and the B cells, I have other images in the paper, but the B cells were actually migrating directly into the plaque of the fibrotic overgrowth. And, it was confusing at first until I found a chemokine that macrophages were secreting. We neutralized it and it blocked B cell migration. So macrophages upstream were actually recruiting in B cells for this response. And that particular chemokine was implicated in B cell maturation in the lymphatics and in the splenic tissue. So what was further interesting is I also found in the literature, which I've referenced in this particular paper, a cancer cell study that also implicated the potential for B cells not to act as almost like an adaptive immune cell secreting adaptively produced and clonally selected antibodies. Instead, it was actually more akin to the B cells were just showing up and secreting cytokines locally, almost more akin to what you usually expect of an innate immune cell.
Right.
And in the cancer cell paper, when B cells showed up, it actually further potentiated macrophages to a higher level of activation state and attack. And what's interesting is that when macrophages are alone at 1 week, fibrosis is at about a level of only 40-50%. But when B cells show up in that second week, the fibrotic level jumps dramatically up to like 90-95%. So you do see further activation, further ramping up of response when B cells engage. And when we knock them out genetically, not just block the chemokine that recruits them, but when we knock them out in a knockout model, fibrosis is stalled at that 40 or 50% mark as well. So we know that they are not the sole. Macrophages really engage the response and precipitate this cascade of downstream B cell recruitment, downstream fibroblast recruitment as well. But they are most definitely important in making it worse.
Yeah, interesting. That's a really interesting finding. Thanks for that.
Yeah, you're welcome. It's a great question too. And so this was the canonical view of foreign body response around the time that I joined the lab late 2011, early 2012, and this came out from David Granger. And so there were early concepts that, neutrophils showed up even first. Everyone always calls, by the way, neutrophils as the first responder. It's a particular point with me with my students because they learned that also from our School of Medicine immunology classes at Hopkins. But they most definitely are a first responder. You see them within the first few hours. But we also interestingly see monocyte extravasation and macrophage recruitment at the exact same overlapping timeline. There were even 1 or 2 other studies, and I'm happy to send anyone the manuscripts of these that's interested in these kinetics. But this implicated that opsonization by antibodies, protein absorption on the surface, spiked an early engagement response. It could also be due to lack of self-recognition receptors, and a couple of other mechanisms that are not quite as well understood. But this schematic, if you saw it, it actually suggests that neutrophils have to be first. And then it implicates maybe someone might logically say, well, if there are no neutrophils there, then do we also eliminate monocytes and macrophages, which monocytes arrive from the blood, they extravasate out into the tissue, differentiate into more tissue-associated resident macrophages, and then they fuse because they cannot physically surround this larger object that do that frustrated phagocytosis process. They actually fuse together into what are called foreign body giant cells to be able to cover the entirety of the surface. What is accurate about this image, though, Monica, is that when you see the monocyte to macrophage to FBGC axis, you see the arrival of fibroblasts at that same exact time. And fibroblasts are typically, while there's been a little bit of implication in the field that macrophages can secrete some collagen, the vast majority of these collagen strands in the background in the ECM matrix remodeling around the device to seal it away is really due to the fibroblasts. And in my 2017 study, I actually depleted neutrophils. And what was interesting is not only did fibrosis occur, it actually occurred even worse slightly. We saw more aggressive clumping. And the reason I suspect is due to there being a subset of neutrophils called myeloid-derived suppressor cells. They act very analogous to Tregs, which are I jokingly call the traffic police of the immune system. They help prevent autoimmunity. They say slow down, don't overreact. It's why we take anti-inflammatories, antihistamines, because even then the immune system can overreact, as we all know. And when we get rid of one of those regulators, one of those negative feedback regulators, the immune response got worse. Interestingly, when I also knocked out T cells, fibrosis also got worse again too because of the Treg loss as well. So it implicates the fact that you need to be very careful when you're using broad spectrum anti-inflammatories or interventions, because if you unintentionally hit these other cell types that make the response worse, not better, you might actually be, as a net effect, increasing the threshold that you have to hit as a minimum burden to actually have a successful therapeutic.
Yeah, so finely tuned balance, just over and
Yep.
over, we're hearing this.
Yep. And so where foreign body response, I show this slide in my class. I wanted to show this to everybody, but this is why does foreign body response even happen? Devices and implants are new, right? Come on, Josh, evolution can't happen that quickly. Well, if you think about it, devices, yes, maybe exist only for the past couple of 100 years, but the reality is I even found an article where there were metallic teeth in a pharaonic king from ancient Egypt. In the article, they made it clear that they did not know whether or not those were in the body, placed in the body during burial preparation, or if that individual had them during their lifetime. But when you go, there's other examples too. So you have shrapnel, lead bullets, going back to bronze, copper age. Believe it or not, if you go back even further into our Paleolithic ancestors, stone fragments, predator-prey relationships with bone and even tooth fragments, wood and wood splinters, and dirt. Even large enough granules of stone and dirt can be fibrous. So this is really a way for the body to just seal things away that it cannot physically extrude and push out of tissue. If it's an organic matter, maybe like a splinter, over time they would break that down. But stone, you can't break down. It doesn't work that way. At least not in our not during our lifetime, not during our lifespan.
Quick question from the chat. What other innate immune cells are accumulating or were accumulating? Any changes in dendritic cells?
Yeah, it's a great question. And so this data implicates it with the biomaterial alone. And and in my experience, there are instances of biomaterial specific phenomena. But I was trying to identify the core axis of what was common between ceramics and polymers, hydrogel, naturally derived or synthetically derived, surgical grade stainless steel, so on and so forth. And really you see the heavy responders that are essential for and needed for foreign body response are macrophages and B cells that make that response worse, fibroblasts as well. We do see neutrophils showing up at the surface. An interesting thing is I told you that when I knocked out neutrophils, the response didn't get better, it got worse. And what's interesting is that we know that neutrophils are most generally associated with being protective against pathogen exposure, parasitic infections, and so on. And so I do believe that this is there are cytokines or chemokines being simultaneously expressed that are bringing these other cell types in parallel, sort of as a more broad spectrum host defense response. And so as I said, once you deplete neutrophils, they don't matter. The rest of this happens just the same anyway. And again, per the data that we had kinetic wise in the other manuscripts, I would again shift that monocyte to macrophage axis backwards to completely simultaneously overlap with the neutrophil response. But in the flow cytometry, what's interesting is that macrophages are the largest dynamic responder. You see them in the largest number. Neutrophils tend to spike at the same early time point, but then they plateau and they remain at a basal state throughout until the overgrowth occurs, and in which case most immune populations tend to dwindle. We associate that with just the body has overgrown the device sufficiently. It no longer sees the surface or the object anymore, and the immune system goes elsewhere. All right, so this is an example of another concentrating factor in the field. And I want to bring this up because we also found this to be this was also at play with this other tolerogenic particle system. And so I don't know, Monica, if you remember, but we found out from clinicians as well as also basic researchers that they would put their implants into biopsy mice. And you can see here, this is a whole lawn of translucent hydrogel spheres. You can see by phase contrast imaging little round circles. There's a little bit of fibrosis throughout this field. Lewis rats in the this is IP intraperitoneal abdomen implantation. We also see reductions in the skin, intramuscularly and other anatomical sites as well. Lewis rat, there's some fur and tissue debris, but most of the capsules are not fibrosed. Black 6 mice, we had early conversations. Dan Anderson brought somebody in from Biogenetic, and we also had a lot of conversations with those in the field. Basically, everyone says, oh, well, anybody knows that profibrotic mice must be C57 Black 6. That is the profibrotic model. Yeah. But into non-human primates and humans, we know that the response is a little bit more aggressive as well. The materials, even in Black 6 mice, this is about a month, or 2 weeks here, but about a month or even 2 to 3 months, the materials start getting stuck into fat pads. We actually see that collagen encapsulated organs like the kidney, the liver, and so on. We've even seen a capsule get indented into the lobe of the liver, but you can pluck it right out and it's not fibrous at all. this collagen capsule tends to seal in the immune system response in those organs. There are millions of immune cells floating around the liquids of your IP space, all around the organs. They're important for preventing bacterial leakage from the gut. They're also important for pathogen exposure and so on, otherwise. But what's interesting is that we see blood vessels vasodilate in fat pads. Fat does not have collagen capsules. And we see, if anything, capsules not only getting individually fibrosed, but we also see them getting stuck eventually in time to nearby fat, which implicates that imagine if you have a high amount of response from a fat pad and there's a hand grabbing out at the material, it's going to get fused to the fat first, if that's where the immune response is actually coming from. And that's what we typically saw. There's a lot of other dynamics. We have videos, like intravital imaging and so on of this phenomena. I do want to get to the main manuscript that I'm presenting today, but I did want to at least tell you about some of the trajectory and the pipeline workflow that we've had over the years, whether it's individuals who focus more on the chemistry side, so modifying the backbone of hydrogel alginate for encapsulation, trying to reduce inflammation by subcutaneous imaging or foreign body response as well. We had a paper come out also on diameter size, showing that the larger a sphere. And we actually found a paper on hernia meshes that implicated that very sparsely woven meshes, very short distances, immune systems can actually bridge those distances and radiate plaque, fibrotic plaque across, but very sparsely woven meshes with very large openings, immune system cells couldn't. And so we were originally trying to rationalize with spheres. Well, is it the curvature? Like if you were on the surface of Mercury versus Jupiter, could you tell the difference? If you could, we could barely see the horizon on Earth, even from a plane. So we figured maybe that's not it. And then eventually we finally figured out from doing surface area and volume normalizations of different amounts of capsules that we implanted, that it was actually a packing density issue. It was actually deficiency it wasn't a material property. It was the fact that the immune system, per that bridging concept, when you have very small capsules, there's very high packing density. Think about grains of sand on the beach. They can traverse those very small distances. Now think about a bowling ball, minimal point of contact, then radiates very quickly away. The immune cells can't physically, mechanically cross those distances, whether it is capsule to capsule or capsule to tissue that even flexes in a little bit. So figuring out new chemistries or new architectures where materials can still gel, we can use different fabrication methods, molding, electrospray, whatever, without just testing biomaterial presence alone, and then with graft on board too, whether they're healthy or SDZ-induced diabetic mice. One of the bigger holdups also in this field, Monica, I'm not sure if you appreciate this too, is that Black 6 mice are profibrotic, and we can induce them being diabetic with a streptozotocin treatment to selectively kill off their beta cells in the pancreas. But believe it or not, there is no autoimmune induction model in Black 6. Now, on the opposite side, you have NOD mice, you have non-obese diabetic mice where you do have autoimmunity, but there is a very compliant, very low or to no fibrotic response in those mice as well, very analogous to the bulb C phenomena. And so thankfully, we've done some cynomolgus monkey work before, was very proud of that work. Everything was sterile survival procedure, both delivery and then retrieval over time with minimally invasive laparoscopic surgery. The monkeys were left alive at the end of those particular studies. But then we moved into realizing that I had a reviewer once say to me, monkey's not a human, so how relevant is that for the clinic? and as cynical as that point of view was, it was technically correct. We do still have species-specific differences. And it stuck with me. I answered at the time, we were testing evolutionary preservation of a core immune mechanism. The second thing was the drug that we identified that my friend Shadi and I crystallized. It was actually a drug, believe it or not, that came from the cancer field to polarize macrophages from being pro-tumorigenic to anti-tumorigenic, and had already gone through multiple phase clinical trials in humans. And so
there's so much that can be pulled from the oncology field to interface with the autoimmune approaches.
Yeah, we got lucky, and then the reviewer recognized the fact, oh, wait, this target isn't human for sure, and it's already been validated clinically, and so they basically said, I get it now, But in order, if you think about that too, and I agree with you completely, Congress has hearings on all of the billions of dollars that have been spent on cancer, and obviously there's very limited progress in terms of the therapy side, with minus a few exceptions such as CAR T and checkpoint inhibitors and so on. But you get to a place of really high level biologic understanding that we would not have had otherwise, and then also development of therapeutics that other fields are now taking advantage of. It's just an amazing resource. So I as complicated as that area is, it's been beneficial in many other ways. I agree with you. One of the things that we recently had come out earlier this year was a paper on humanized mice. And so we recognize the fact that monkeys are prohibitive, they're costly. You sometimes need dedicated, highly trained technicians to work with them too. And think about the limited numbers of them as well. Ideally, you can only be so invasive in a monkey or even a human. Whereas humanized mice, when we irradiate the bone marrow, we inject in human cells to repopulate a human immune system instead of a mouse one. We can now be as exploratory as we want to be as though we were testing in just other mouse strains. And furthermore, we actually, in this particular case, I would argue in some cases it's better than monkeys because you now get to study human immune cell interactions and dynamics with your therapeutic directly.
Agree.
Yeah, one disclaimer is that there are many variants of humanized systems. We actually tried 2 or 3 different versions of them and could not get fibrosis. And the irony was, Monica, is that we had really great, after doing flow cytometry and with my picking apart the wild type Black6 model system, we realized that the humanized variants early on had really good T and B cell levels, but their entire innate arm of the immune system, including macrophages, was null and void. And so in order to get fibrosis to come online in this model, we had to elicit 3 additional human cytokines delivered into the mice, and they enabled both the proliferation as well as the maintenance of the innate arm of the immune system. And now we get fibrotic responses. Very cool. All right, so this is just a quick example. Black6 mice, it's the same background strain. It's cleaner than comparing NOD to Bulbc to Black6 to NSG, all these different background strains. You want to keep all the other genetic backbone the same and really only change one gene at a time, whether it's knocking it out or using a transgenic. And thankfully, the Black6 model's been around for so long, and we also benefited from being the profibrotic model, that we could utilize versions of these strains. And here are the mutations here, if you want to know about them. But a T cell alone, knockout B cell alone, both combinatorially getting down to the level of innate natural killer cell, depletion, as well as even macrophage and dendritic cell dysfunction. They technically exist in these mice, but they do not appropriately mature in response to cytokine stimulation, so they're somewhat dysfunctional. They're also in wild type mice. There's mafia mice where the macrophages have EGFP labeling. You can induce depletion in there as well. There's clodrosomes that target macrophages in wild type mice, fully wild type, so there's no weird selection of an inbred strain with further genetic modifications. To deplete neutrophils, as I told you, we actually use an antibody-based depletion method. Here's that chemokine that ties macrophages to B cells, and I use an antibody also to bind and to neutralize it. And so it could not bind its downstream receptor. It was pepper-coated with other antibody, and its amino acids were not available to bind the receptor where it needed to bind. Yeah, and so only in the case really of when we got rid of macrophages, either alone as well as in combination with neutrophils. Here's the example. Here's a wild type response at 2 weeks in the mice. This is actually with neutrophil depletion. You see these other little tiny clumps of capsules. So there were other images that had all 5 of the mice. I put all N of 5 images in the supplement of the paper so you can see the spread of the data. But we had these extra clumping events that actually kinetically occurred much earlier than we typically see them in just wild type alone. And here all the capsules were with macrophage loss. So it was quite abrupt in terms of what we saw.
Yeah, striking. That's a striking difference.
Yeah, which ones were definitive versus not, right? And then so that made us focus on, even though B cells were interesting, I put in green here, oh yeah, there's about a 40 or 50% drop in both these cases. It wasn't getting rid of it fully. So then we focused on the macrophage axis specifically. Here's monkey data with the omentum in the abdomen, where we put the capsules, and within a matter of only even 1 to 2 weeks, this is at 4 weeks, the capsules were starting to sink in and disappear into the fat-laden connective structure that wraps around the GI tract. It doesn't exist in mice. You actually open mice up and you see what is normally drawn in an anatomy textbook. In monkeys and in humans, there's this fat infused and highly vesselated, you can see all the blood vessels here, structure called the omentum. And it's actually important for, if you've ever heard of the expression that fat is important for cushioning our organs, the omentum is a part of that protection layer in the body. But this being very fat laden and not having a collagen capsule, all of a sudden you now see loss of resolution of the materials as they're sinking into this fat work and fat lattice work. And so this is fat adipocyte tissue. It tends to look like a spider web when you do sectioning and in histology staining, because adipocytes are about 50 to 60 microns. Most of that volume is filled with a big lipid vacuole, which gets washed away. And so the nucleus is pushed all the way over to the membrane. And so you mostly see the membrane lattice work with an individual nucleus pushed off to the edge. And minus a couple of blood vessel structures too. When you look at the implanted omentum, we actually were able to, believe it or not, pinch off this omentum, tie a lariat suture around that clump of pinched off tissue, and then we snipped it with a laparoscopic snipping tool so that the monkey did not bleed out, and we kept the animal alive following even collection of these tissues. And what you're seeing here is that not only are there capsules all throughout the tissue, there's very dense blue collagen in the mesotrachome image throughout and surrounding. It's completely remodeling the tissue. We know that fibrotic tissue really can be irritable, irritate and itchy for the patient. This is immunofluorescence staining showing macrophage presence around capsules in the monkeys, as well as also yet again alpha-synuclein actin showing a fibroblast response. Having worked actually, believe it or not, Monica, in cancer immunology for my PhD, I learned how to dissociate whole tissues into viable single cell suspensions to stain and run for flow cytometry. So I modified those techniques to be able to dissociate cells off the surface of the biomaterial surface as well without destroying the cells. And whether it's black 6 mice in the top row, you're looking at mock implants, so very low background here, versus implanted. So the x-axis is standing for C11b, it's a general myeloid marker. The y-axis is standing for CD68, so that's a macrophage marker. So if it moves in the x direction as well as the y, so basically into the upper right quadrant, you see this red circle highlighting double positive macrophages. And we see single positive C11b in this particular case because they're CD68 negative. These are neutrophils down here in the bottom. If I had used LY6G, you would have gotten an inversion. The smaller population would have risen, whereas the macrophages would have stayed low. And I was also able to do very similar staining in the non-human primate model, and we got very similar magnitudes and kinetic waves of response between them. What's further interesting is that the target that was identified based on perturbation and next generation drug target identification was colony-stimulating factor 1 receptor. It was the focus of this particular paper. A reviewer asked, actually, is this on the individual punctate macrophages that are infiltrating through the tissue, or as well as also on the foreign body giant cells, or was it only on one or the other? I found a paper from the late 90s that implicated it was on both, but doing immunostaining, you can see not only the individual macrophages before they fuse, you also see the fusion events. You see these large swaths of positivity. So there's a little bit of autofluorescence with the hydrogel capsules. There's a little bit of separation between the tissue and the material, but the tissue ring around the surface of the biomaterial, these are all foreign body giant cell fusion events. All right, so using this drug target, we crystallized it. If you don't have any drug on board, fibrosis occurs, it diminishes. Everyone thought that it was the capsule that blocks diffusion and the grafts just die. But believe it or not, the kinetic wave of graft loss was actually corresponding a little bit earlier to really fibrosis being fully overgrown, fully overgrowing the material. And one of the ways I want everyone to think about this is that immune system cells, if there's a massive wave of response, they each in turn are also taking respiratory and nutritional demands. And so if you have a wave of live, vibrant cells that are highly proliferative, and there's a lot of data on macrophage and even T cell responses and how they're different with metabolism, immune metabolism. We know that they're stealing nutrients away. They're outcompeting the graft from ever getting nutrients. They're not going to diffuse all the way through. But when you get rid of this wave, whether it's a liquid dispersed drug, the drug will eventually run out. Eventually, the graft will be lost and you'll revert back to hyperglycemia. In the case of a controlled long-term delivery system, we can really retain drug for a much longer period of time as well as cures. And we had, again, in rodents over 1.3 years with islets, human islets, we had good efficacy too, in addition to rat islets. And in monkeys, we had release over 6+ months. So getting to the crystals, though, just to show you what this looks like, the gliadel wafer system was one of the first FDA-approved polymeric encapsulation systems. It's a tortuous network of open channels where drug is contained within, and they just by Brownian motion bounce around and eventually are released. And so this was actually originally innovated also yet again for cancer. Where they resected a primary brain tumor. The tricky thing about glioblastoma typically is that there are individual cells that move past the margin. When you remove the primary tumor, they come back and repopulate and regrow the tumor. And so there's very poor prognosis in that disease. What's interesting is Henry Brem, who's the head of neurosurgery and science here at Hopkins, co-developed this wafer system, actually with Bob Langer, who was my postdoc advisor, back in Judah Folkman's lab at Boston Children's Hospital. And they utilized this system to deliver anti-proliferative, anti-cancer drugs to hopefully kill those cells that had exceeded the margin that were not surgically resected. Well, we tried using this platform as inspiration, putting the drug into polyurethane, but the drug was so hydrophobic. A lot of these receptor tyrosine kinase inhibitors are very hydrophobic. If you want to deliver them, you have to deliver them daily, and you usually have to dissolve them in DMSO, a large fraction of DMSO, which is an organic solvent. It'll dissolve tissues. And so that's like a pharmacologist's worst nightmare. Some of these drugs are given in pill form orally, but the bioavailability is reduced as well. So the first instance, the drug was not compatible, did not end up in the polymer. The second one, it did end up in the polymer, but then it didn't want to let go of the polymer because of the hydrophobicity of the drug when it was placed into aqueous solutions. And so my friend Shadi, who is now an assistant professor at chemical engineering at the Tech now in Haifa, Israel, he came into the lab and originally had created these nano-layer thin coatings of drugs on stents, and they were with anti-proliferatives, to prevent plaque formation so you wouldn't get stroke. And I challenged him and I said, well, can we take powder drug from the vendor? And here's a 50 micron scale bar by SEM. And can we grow these hundreds of microns to millimeter scale crystalline depots instead of nanolayer, mega depots, basically, for long-term release. And so this is a 200 micron scale by comparison. So these are literally hundreds of microns to millimeter scale. When we put them in the hydrogel, you can see the line of the hydrogel is retained because a pocket opens up as surface erosion occurs, layer by layer by layer of drug is released slowly over time. And we ended up publishing that work in 2019. This is actually with the crystals in green. So glucose levels were reduced at first, even with regular transplants with no drug, fibrosis would ensue. And usually with that massive wave of cellular response, within about 30 to 40 days, typically you see death of the graft and loss of insulin secretion in response to glucose, and you have reversion back to hyperglycemia in the control. When we put in the drug in an amorphous powder state, it did elongate cures by about even 30, 40 days. And that is what has usually typically been seen with a lot of other drug elution systems that usually can go anywhere from 30 upwards of maybe 90 days, typically. What ends up happening, though, interestingly, is that once the drug runs out, the immune system, through new biogenesis, creates new cells that have never seen the drug, they're not inhibited by it, and all of a sudden they engage the response later down the line. So trying to get this release system to release as long as possible was very important. And the crystals, as I told you, in the green case, we had cures for over 1.3 years in mice. And when we took the capsules at the end, there was still drug within. So this data is new. It's not published yet. And so we have new project funding from the JDRF also looking at this phenomena. And what's interesting is there's been a lot of work on human cells, iPSC, embryonic cells, or even like you heard about the pig heart that went into a human. So there's this alpha-gal knockout too for transplantation. What we do appreciate now is that when you have biomaterial alone, and I've simplified this a little bit, there are other immune system cells involved too, but macrophages and B cells in red and green are the first two that I've shown you are largely responsible for biomaterial response. Dendritic cells and natural killer cells are two of the cells that come in with graft response. And when you have blank, empty capsules, you see basically, you can normalize that to a level of 1. When we put syngeneic mouse islets from the same strain into the capsules, it looks no different than capsules alone. You don't see really strong engagement of any of these other cell types. When you go to more xenogeneic cell sources, such as rat, and then pig into human, you actually ratchet up the amount of rejection response you see. And so in the bottom case here, the images where we can prevent fibrosis of capsules alone, we can prevent allografts with mouse islets with capsules alone, with our antifibrotic drug. Rat islets actually work reasonably well with our drug, but we did show a little bit of there's actually a thin film of growth, believe it or not, still on the capsule, even though the graft is still permissive in the model. It'll survive better, but there's a little bit of immune response there, and we're trying to now appreciate what that is. With very xenogeneic pig or human cells, though, the response gets significantly worse. And we did show data where our crystals work for human islets outwards of almost 3 months, but it did not go the distance that the rat islets did, which was over 1.3 years. And so where there is this ratcheting up of rejection, the further away you move from the host recipient. This is also data that's in submission. This is actually allogenic islets coming from a monkey with our antifibrotic crystals. And you can see with capsules, really great maintenance of viability across most of the set. But when you go to either primary derived human islets or to SC beta cell, so iPSC derived beta-like cells, you actually get very heavy loss of viability, even with our onboard antifibrotic. So we now know that there is an additional immune mechanism, even when you're preventing fibrosis of the biomaterial. It's not solving every case. If you're only doing allogeneic transplantation in humans, maybe that's fine. But a lot of cell sources, a lot of organ sources are coming from other animals, other people. Humans are not genetically as streamlined as all these really highly inbred strains too. We even had a conversation about the monkeys that we're using, and we realized that even though monkeys are more diverse than highly inbred mouse strains that were inbred going back 15 or 20 generations, most of the monkeys we were utilizing, shocking to also learn this too, is that they mostly came from the island of Mauritius. And so that, if you know anything about human genetics, humans coming from the island of Iceland or in other isolated genetic gene pools, they're going to be a little bit more genetically bottlenecked as well. So is it really fully genetically diverse the way a human gene pool is? Probably not. All right, so just to orient you a little bit further, dendritic cells are part of the innate immune system. They are one of the largest antigen, they're one of the 3 antigen processing cells, along with macrophages and B cells, but they're one of the largest, they're largely responsible, I would say, for not only processing, but then presenting antigen to adaptive T cells for stimulation for many different subsets, as well as also B cell stimulation for producing either general antibody response, so monoclonal versus polyclonal response as well. All right, so this really brings me to the new work that I want to present today. And I know, and that was quite an extensive background of the field, I think, where it's come from. Cell encapsulation is as old as I am. It was published in Science in 80 or 81, and I was born in 1981. So this field stagnated for a long time because of fibrosis, because of these issues, because of the strain differences, having divergent responses in the immune system just based on how they were randomly, selected for. And so there was an earlier paper coming out of Jordan Green's lab where they had tolerogenic APCs that were made from a PLGA polymer core. I'll show you an image of this, as well as a PBAE blend. And we modified it. So they showed in vitro, they actually isolated T cells from the spleen of black 6 mice and showed that they could stimulate naive T cells into turning into selectively Tregs. And so the concept in the modern field is, do you need biomaterials to prevent fibrosis? It's complicated. People, even the field has moved away from microcapsules towards a singular macroencapsulation system where they can guarantee retrieval of the entirety of the graft if they have to. But even in those cases, this indirect diffusion mechanism is still a problem. So these tool sets, even though they've not been perfect therapies along the way, they've actually been informing us also about the biology, and that it's not just biomaterial response now, it's also this indirect graft rejection response. Now, the theory in the field is that can you induce a microenvironment of immune tolerance? So there's Tregs as an example are important for preventing autoimmunity normally. They're one of the major parts of our peripheral tolerance mechanism where they keep the immune system quiet. CD8 T cells can kill cells directly just on their own. And so Tregs are there to basically prevent that from happening normally. Obviously, when you have autoimmune disease formation, things go awry. All right, so we were able to apply these particles and modify them in other ways in order to use them in vivo and to actually show not just in vitro stimulation in the context of co-culture with islets, specifically for type 1 diabetes, but then also doing co-injection with islets into the in vivo tissue. And we did injection into the skin rather than the renal kidney capsule. So that's something for a future direction. But this was a proof of principle concept that we were very excited about. And so the idea here is that encapsulating biomaterial takes up all this extra volume. You have foreign body response to it. Can you use particles to just recondition the microenvironment, and hopefully that there's an immune memory to that reconditioning, and then you can then inject a graft into that site, and then it's permissive. You don't have immune rejection. There is a question I do want, as we're going through this, there is a bigger question in the field, though. We do have some diabetic mouse data, but it's not autoimmune diabetic. It's STZ black 6. So there is a question in the field that when you have autoimmune diabetes and the inflammatory response is a little bit different because of that immune dysregulation, would these particles have the same effect? And that's another question that we're looking at too. So the way that these are produced is that there's basically an oil water emulsion system. Eventually, we use solvent evaporation as a second step to just get the PLGA particle core. And then they are functionalized with surface chemistries to add a number of different entities, including antibodies that I'll show you in the next slide for stimulating T cell response. We get similarly sized particles with both the PLGA base material as well as the PBAE blend. And you see that here as well, also by dynamic light scattering. Zeta potential does change a little bit for both of these particles at a slightly different pH, but they're typically quite standard. All right, so what we're conjugating to the surface, and I'll show, I think on the next, I thought I had another image of it, I apologize. I think it's on the later slide. Actually, let me show you that first real quick because that's easier to understand. So here is the PLGA or the PBAE in green blended core, PLGA in blue. We add an anti-CD3 and an anti-CD28 antibody. We know that both of those antibodies together have been used on Dynamax beads and so on to stimulate CD8 effector T cell response. However, if you add TGF-beta, which is a tolerizing agent, inside of the particle, you actually skew response towards CD4 positive, FOXP3 positive Tregs instead. All right, so coming back to that data now. So here is a data set on the bottom left where we see the fraction of FOXP3 cells out of all total CD4 cells. And this was studying a ratio of how much anti-CD3 to how much anti-CD28 do we need on the surface to get maximal skewing of T cell activation towards Tregs. And so we eventually saw that the 5-to-1 ratio here was most efficient. So that's what we basically eventually maintained through the rest of the study. There's additional data here on conjugation efficiency being comparable and other outputs as well. But I bet 5-to-1 typically perform quite nicely across the board, and that's what we resorted to use. All right, so early on, I showed you bulb C versus black 6 differences to foreign body objects, large objects, macroscale objects. Well, believe it or not, we're also seeing interesting differences here from bulb C to black 6 at the level of now microparticle injections. These are 1 micron scale particles and even nanoscale particles in our earlier studies from the Green Lab. So in the left-hand case here, interestingly, when you have no conjugation on the surface, there's no antibodies on the surface, you see a little white particle spot. Maybe there's a little bit of an immune clearance response there. What's interesting is that when you conjugate them with all these signals, you not only get a large white infiltrate zone, you can see that large globular white zone here, but you also get a dramatically increased vasodilation and branching of all the blood vessels under the skin. Actually seems to be quite inflamed. In the black 6 mice, by comparison, with unconjugated, the response looks quite similar. You can see this little tiny white pellet zone. With conjugation, you get a large immune cell infiltration event. There is some vasodilation of the blood vessels, but it does not appear to be as extensive in the bulb C mice. Now, furthermore, when we started to do gene expression, we were looking at the major markers of Tregs, FOXP3, as well as cytotoxic T cell CD8, because we wanted to know how were these particles going to skew T cell activation, not in vitro, but in vivo. And in the bulb C mice, we eventually saw not much occurring locally at the injection site. What ended up becoming much more indicative of maybe a more pro-inflammatory response was in the bone marrow of bulb C mice, going from white here to blue. We saw a higher upregulation, higher upregulation of potentially FOXP3, but it was not statistically significant. So it was not having the local intended effects that we were seeing, despite this vasodilation response. And so we eventually moved away from bulb C towards black 6, because when we looked in the black 6 mice, we actually saw a favorable response, which I'm going to show you in a second. There's a question here. Please. Okay, so you said feel free for questions. So again, this is now an image showing you bulb C on one side, black 6 on the other, profibrotic on the right. And just know that typically why we now appreciate this is that black 6 are known to be more Th1 innate immune heavy skewed. Bulb C mice are more Th2 adaptive immune skewed. They're heavily used for cancer vaccine trials for getting very high antibody titer response. And as I've shown you, we don't get much fibrosis, whereas black 6 we do. All right, so here's more particle response. What's interesting is that there was excellent with PBE conjugation. Furthermore, we also got the Green Lab, in the earlier study, got really good interactions with T cells. So this is interaction with cells with the APCs themselves, with higher amounts of the total APC dose. So FOXP3 is on the x-axis. You see that PBAE was more effective at increasing Treg activation in vitro. And why they associated this, or why they assumed this was the case, was because that the PBAE particles, believe it or not, through this combination chemistry, they were actually more sticky. They were getting higher amounts of protein absorption on the surface, or more antibody conjugation, conceivably leading to more Treg activation. And so here's an example of where at one particular dose, the higher dose here, you really see a much higher amount of FOXP3 plus CD25 plus helper Tregs. What is observable yet again is in direct comparison, though. Here's the PLJ PBA blend. For in vitro FOXP3 activation, it's much higher than without any APCs alone, and also with just the PLJ core. And this was not just in numbers. So when you're looking at immune response, you don't want to just look at the numbers in the tissue. You also want to look at their phenotypic state. Just because you're FOXP3 positive, it's actually been heavily debated in the field now. You cannot only get away with showing FOXP3 data. You have to actually show that not only are they markers are showing identity that they may be Tregs, but then you need to actually dissociate the tissue, take them out, do something else that actually indicates functional changes, phenotype changes as well, to show that they are in fact inhibitory or suppressive. And that data is sort of graphed in two different ways. I think it's a little bit more clear here on the right-hand side. This is actually percent suppression when we had a higher amount of suppressor Tregs to responder cells. And you can see with the PBAE blend, there's a higher amount of overall suppression as you increase the proportion of quote unquote the Tregs that we had in vitro. All right, so moving beyond the earlier in vitro work that the Green Lab did, there's going to be some very interesting differences in vivo. And we assumed, well, PBAE is more sticky, there's more protein on the surface, it's going to lead to higher Treg activation also in vivo. Well, that's not quite what we saw, and it was very surprising, but I'm going to get to that. It still did work with the intended effect that we wanted, but it was a little bit different ultimately. Is there concern when the TGF-beta runs out? Yeah, so there's actually concern. So these PLJ particles in general are biodegradable. It's actually an excellent point. I'm going to come back to that loosely at the very end when I show you the in vivo efficacy data, because we have some very short-term partial responses that don't last, that they don't stand the test of time. And I'm going to talk to everybody about that at the very end. So here's some distant tissue just to orient you for H&E staining and Mason's trichrome. Again, we usually use these two stains for cell infiltration and fibrotic type responses. With the PBAE particles, what's interesting, sorry, the PLJ unconjugated, you actually get this sort of injection zone, this immune-laden response, which we think is more akin to potentially foreign body response. There's no protein on the surface modulating the immune system, so they're just only responding to the biomaterial. What's interesting is that when you do conjugate, whether it is in male mice, we tested both males and females, or female mice, we actually see a morphologic difference in this infiltration zone. You actually have an outer lighter corona, as well as a darker inner core of this infiltration pocket. It's very interesting. Now, when we skewed more towards the PBAE blend, the outer lighter zone tended to disappear. We saw that maybe a little bit of this outer lighter zone here with the unconjugated, but when you conjugate fully and you have TGF-beta, most of this infiltration pocket is now a very dark, dense color. And we suspect, and this is across males and female mice, and we suspected that it might be due to more effectively throughout the pocket, creating a more advantageous microenvironment, but we're going to show you some data on that just now. So as I told you before, we hypothesized that conjugation, even with PLJ, would solicit higher Tregs in vivo. And what was interesting is that we actually had a problem in vivo. And the problem in vivo was that this response to PLJ by itself actually created a high amount of Treg presence. And why that is the case, I suspect, is that when you have usually a pro-inflammation response and you get an increase in CD8 T cells, and we see that here, there's a similar amount of CD8 T cells to the conjugated in these two cases. The body by itself upregulates Tregs to control that increase in CD8 effector T cells. Otherwise, you're going to have the chance of a lack of tolerance in the body. With the PBAE blend, so we suspected, with more protein on the surface, it would be even higher activation. Well, we didn't see that. Surprisingly, we saw the opposite. We saw, technically, statistically, there's no significant difference between this level in blue to level in green. What was most surprising, though, is that the base blend material had a much lower inflammatory response. And it was not just at the level of FOXP3 Tregs, it was also with much lower CD8 T cells as well. Importantly, unlike with conjugated PLGA, you don't see increase of CD8s with conjugation. You only selectively see increase of FOXP3 Tregs. And so when you're graphing a ratio of the two, because they really do interact cell to cell, so taking into account how many of one versus how many of the other is very important. When we take into account the slight ratio, there was a slight increase with PLGA, sorry for the slight offshift here, because of a little bit of reduction in the overall mean amount of CD8. So the CD8, if the denominator goes down, the overall number is going to rise, right, mathematically. But what was very clear night and day is across both male and female mice is that we saw much more advantageous FOXP3 to CD8 T cell ratios when using this blend particle system. And so the mechanism of us getting there to a more advantageous state was a little bit different than the conclusions you might say from the in vitro findings. In vivo, things work a little bit different sometimes, but thankfully, it ended up being the same overall net effect nonetheless. This is actually a much more complete panel, not only showing you FOXP3 and CD8 here on the left, also shows you very high upregulation of CD4. We see a little bit of neutrophil response, maybe a little bit here of CD74 and even C11C not on this plot for dendritic cell response too. But overall, most of the immune populations, it depends if you're comparing distant skin or just base level material unconjugated versus conjugated in terms of having responses. This is in the manuscript. I don't want to harp on it too much, but we still at least saw very positive overall from a cumulative response of the immune system. What we were seeing, we liked. Now, what was also interesting is that indication of increases of CD8 T cells was apparent based on immunofluorescence microscopy. Here's CD3 staining following particle injection unconjugated PLJ versus conjugated. Here's FOXP3, but here's also CD8, and that's not ideal, right? What was much more interesting to us is that the unconjugated PBAE had very low background, even lower CD8 background than the PLJ by itself. But importantly, when we injected with conjugated particles, we had very good FOXP3 levels and lower, dimmer levels of CD8. And this again showed us overall that we had skewed the T cell response at least in the right direction. All right, so There's a couple of functional assays that I want to show you guys in vitro real quick before I come back to the final in vivo efficacy data. So this is actually, we called it a tri-culture assay. So this was us taking, the team took spleens from black 6 mice, and they isolated CD4 and CD8 T cells with isolation kits. In addition, we took islets, we derived primary islets, not from black 6 mice, from bulb C. So going across strain of mice was a way for us to try to mimic allogenic, different individual, same species, but being a little bit more genetically distinct, a little bit more different. Like if you were taking islets from one human and putting them into another, I have a twin brother, so maybe it's less of an issue for me, obviously, although if I have autoimmune diabetes, chances are he might get it too. But in terms of transplant grafts, this is a better way of at least spiking. We know that there will be an immune response to the bulb C-derived islets from the black 6-derived T cells. So we use these naive T cells alone first, and we culture them either against a control well that has a CD3 and CD8 and TGF-beta. CD3 and CD28 were conjugated to the plate surface with TGF-beta in the solution, I believe, or we incubate the cells in presence of different milligram amounts of the toll APCs. Following activation of those cells, they then get all cumulatively CD4 and CD8 thrown onto the islets at different ratios. So the first number here is CD4 to CD8 ratio. So here's a CD8 alone control where we have no CD4 cells. So obviously the ratio of CD4 over CD8 is 0. When we use the control wells, there's reasonably good skewing, especially with higher amounts of CD4 T cells towards positive response with control wells. But we also see a dose-dependent effect ratcheting up the toll APCs, and eventually we get to reasonably good, similar efficacy as well. This, by the way, was just looking at general CD4, not FOXP3. So we're going to actually show you more significant data that's actually going to implicate the toll APC particles work better than this plate conjugation method. But before I do, how we identify the cells, we also used assays following in vitro tri-culture incubation. We took the islets and we dissociated them. We trypsinized them apart. Got this protocol from Jeff Moman's lab at WashU St. Louis. So Jeff, if you're on, I thank you for that. Very simple, very, very passive protocol to just dissociate the islets and run them through a flow cytometer. So we gate on all cells, we get singlets. This error should be down here. I just cut and pasted last minute, I apologize. So these are singlets of, we can gate out CD4 and we can gate out CD8, and all of the CD4 negative and CD8 negative cells that are remaining were all of the alpha, beta, gamma, delta, and so on cells that came from the islet clusters themselves. And then once we look at those specifically, we use a live dead stain to indicate which ones are alive versus which ones are dead. Now, what's interesting is using the same exact in vitro condition, showing you a few more control conditions, is that the CD8 alone had obviously a higher amount of killing potential. So there's higher amount of dead islet cells. More helper cells to fewer helper cell ratio, you actually see less death to more death in a dose-dependent manner. And what's interesting is so now showing you efficacy, the toll APCs in vitro work quite well across the board of protecting these islets, even at ratios of 1-to-1. Not necessarily having to rely on a heavier ratio of 2-to-1 for helper T cell to CD8 T cell. Yes, yes, clap. Yeah, please, props to Jeff and his team. They're amazing. And here's just some images also of live stain that we did. This was a little bit more subjective, just trying to do some immunofluorescence microscopy. What is clear, I think, is from the 1-to-1 ratio of the control coated wells is that you have less biomass of material versus the particles in all cases, especially at the same 1-to-1 ratio here and here, which was really cool. And then, but lastly, we really wanted to test the function of these cells, not just the viability. You want to look also at insulin production, right? So this is a GSIS, glucose-stimulated insulin secretion assay. Here's the control level. We normalized to 10 islet equivalents. Islets alone going from low glucose to high glucose back to low glucose again. You can see with CD8 T cells only, there was high killing of these grafts and very low insulin to no insulin production. With the control coded wells, there was a little bit of a rise, but it wasn't statistically significant. And interestingly, only with the toll APCs, and in this case here, these are the two higher doses. Even with there were a few islets in these wells. We were trying to do so many conditions. Just always be mindful of that. When you only put so many islets into a well, I think we did one experiment. We did 20 islet equivalents, another one where we did 50 to try to increase the number further. And that's why we normalized based on 10 islet equivalents, because it wasn't the same across the experiments. But what you're seeing here is that definitely in the high glucose case, there is sensitivity and there is statistical significance in terms of being higher than just the basal level background of the assay. All right, so moving forward into an in vivo experiment, we did the similar thing of taking biopsy islets. We mix them with our toll APCs without any preconditioning whatsoever. So as opposed to taking naive T cells from a spleen, preconditioning them first and then throwing them into a well. This was just mixed toll APCs with biopsy-derived islets and throw them into an STZ diabetic black 6 mouse model. And we saw a number of interesting things. So with islets alone at day 3, you have higher spikes and some immunity responses here. At day 7, when there's more clearance of the graft, the immune system, we already think kinetically is starting to dwindle. What was quite interesting is with the toll APCs, even though yet again, there was a little bit of a CD8 response and a little bit, you can see magnitude-wise, it's a 6-fold change. FOXP3 increased to a dramatic, almost 150-fold level. And so yet again, with the ratio of FOXP3 over CD8, there was a much more advantageous microenvironment reconditioning in the toll APC case, at least at day 3. So per the question that was received, I just want to get the name again. I think it was Lucy. Thank you, Lucy. So this is, we know that from talking with Jordan and his team, we know that these PLGA particles tend to degrade over a course of about a week. And unfortunately, what is interesting is that we see loss of this induction given time, but it's a little bit more complicated than that. And so what this begs is that we have to engineer the particles further, either make them degrade slower, put these factors onto a gel system where they're retained for longer. We've talked about these elements in the discussion in the paper, but now this is actually blood glucose data from diabetic mice. And what you're seeing here, believe it or not, in the combination group in blue, in teal blue, is with islets alone, there's a little bit of a drop in glucose levels following induction of diabetes. The problem is that effect is lost quite quickly once those grafts are rejected within a matter of only hours to days. What we see, interestingly, with the toll APCs is that not only do we get a similar level of drop, it actually is retained for about another 3 to almost 7 days longer than the islets alone, from about here, day 4, outwards to maybe about 4 days or 5 days here to day 7. And I want to bring up 2 issues. So one of which is the biodegradation of the particle, which I've already mentioned. But another thing I also want to mention too is that when I showed you this immune data before where it's specifically focusing on T cells, we know that these particles work to protect the graft in vitro when you only have T cells by themselves, but that is a very artificial premise, right? We know in vivo, there's the full broad spectrum of the immune system. And what was quite interesting is that we have data here implicating dendritic cells as well as natural killer cells in the response against the graft, which implicates that we can not only engineer these toll APCs to only eliminate T cell response. We have to have something that is a little bit more broadly reaching, whether it is at the antigen presentation level or at an innate lymphocyte level too, natural killer level too. So really, really cool mechanistic data that we're discovering here. The particles for generation, I would say 2.0, because they were after the in vitro efficacy work that the Green Lab had done. We're now showing in vivo efficacy data with a couple of other considerations. They have to be engineered further, hopefully for longevity, as well as more breadth in covering more cells of the immune system. So with that said, I really want to be grateful. Jordan gave me his consent and permission to present this data to you all today. He's traveling right now with one of his kids. He's doing the college circuit. His children are getting to be college age. Sarah Neshet is actually a co-advised PhD student across both of our labs. It was very forward-thinking, but she recognized that she wanted both a senior as well as junior PI interaction for her PhD experience. Stuart Bauer is one of the other co-authors of the study. He was originally a master's student. We were very fortunate enough to keep him as a PhD student moving forward as the lab was developing early on. Kelly Rhodes has since left and graduated from the Green Lab. She was heavily responsible for that earlier biomaterial study from 2020, but she was also contributing to the study as well in a third co-author capacity. Gina Wong, Victor Quiros. Gina's graduated. She's now at Novartis and IBR in Cambridge, Massachusetts. Victor was an NIH prep scholar who stayed on as a PhD student. We were very lucky. He did very incredibly well in his interviews, and we were lucky that he wanted to stay at Hopkins. And Stephanie Zhang is a research assistant professor in the Green Lab as well, also contributed to this project too, along with Justin Wong and Shirley Lemaster. So others in our labs, this funding was largely from the Juvenile Diabetes Research Foundation, so I very much want to thank them too. Our lab has done well over like I said, we're here for almost 5 years. For the past few years, our graduate students have also been very productive, getting graduate research fellowships as well. And I want to give them a lot of props on that and assisting the lab in every way that they are doing too. So with that said, I'm happy to take any questions you guys have about this work or about just the general field of diabetes and what people are attempting to do now as next steps.
Josh, that was just a great talk. It was a fantastic that you framed your data with the historical sort of lead up of the field. And it was just really, really beautiful. So I thank you for that talk. I had two questions.
Please.
Anyone else can drop their questions in the chat, but I have two sort of big picture questions that pertain to combinatorial approaches. So the first is, you tapped on sort of, mental environment, and it's the patient as a preferred implant site.
Yep.
Obviously, there's proximity to the intestine. Emmer Altinus lab at Boston College. We saw that we had some of his people on here. They're working tirelessly to decipher the microbiome changes that accompany autoimmune diabetes. Do you think there's room to investigate and ultimately coordinate microbiome contributions to mitigate fibrosis in islet implants? And if so, what might that look like?
Yeah, I mean, I've reviewed studies that have been looking at this, and there's also work coming out from Jennifer Liseff's lab. There's multiple groups trying to look at effects of microbiome. I would say that one of the bigger concentrating variables that I've seen thus far is that we know that germ-free mice that don't have microbiome, are immune dysregulated. So we know that the GI tract is so large, it houses approximately 60 to 80% of our immune systems. And that in particular, those immune cells don't just, some of them stay there to prevent bacterial leakage, as I told you before, but many of them actually move to other tissues after they're educated to have effects in other ways, in other disease states, and other anatomical sites. So, some studies, when you're using germ-free mice, the question is complicated to deconvolute the fact that there's no microbiome and the immune system is also dysregulated, right? We know that if you just inhibit the immune system by itself, you can reduce fibrosis. So it's complicated. In wild type systems where you have a microbiome, people then try to use antibiotics or other interventions to knock down the microbiome or knock down microbes. But everyone always thinks about antibiotics as being prokaryotic selective, and they're not. They actually have some major eukaryotic and specifically immune cell side effects, which lead to loss of macrophages included, and that will lead to reduction of foreign body response. So I haven't seen a premise really yet showing me definitive mechanistic data really hammering this point home, because these two, either on the depletion side or on the maintenance side, they're both in concert with each other, affecting one another. There's this feedback loop between them. But it's really interesting. So why we did work in the abdomen or the IP space, believe it or not, early on, we were actually challenged on why we were operating the abdomen. Everyone always said to us, well, that's not the traditional site for biocompatibility assessment, which is the skin. People just easily throw things under the skin because it's easy access and it's less prohibitive.
But it's not as vascularized.
Completely agree with you. And I would actually argue the opposite. I would actually say that the skin, if your anatomical site of use is in a different tissue, test your technology in that tissue directly. Do not just rely on the skin, because you're going to see different things. You just will, and that's one example of it. The reason why we're going into the abdomen, though, is because in order to truly reverse diabetic state, like in this case here, we didn't. We actually did inject 500 islet equivalents, and we actually injected them. We split them and the particles into two different spots, half on the left and half on the right. And based on the capsule experiences I said, don't just inject them in one clump. Create a needle track, push the needle in further, and then push the syringe as you're drawing the needle back, almost to make a line under the skin to try to spread them out further, because you said it's less vascularized. We don't want to crowd the graft. We know that the skin has historically, as a consequence of these issues, been less hospitable to graft viability. Right. So that is another potential variable here too. A reviewer said to us, well, why didn't you go into the renal kidney capsule? Well, we were hoping for an easy access point in the skin. That would be like the holy grail of getting this to work. We did get the crystals. We did get the crystals in the capsules to work under the skin, because we got rid of the wave of the immune system, and so the remaining amount of oxygen nutritional flux or diffusion was not limiting. Interestingly, however, it wasn't the case. It wasn't really the case here. I would say that yes, testing in other anatomical sites is also a very big variable. Abdomen was useful because you have more vascularization in the fat of the abdomen over the fat of the subcu space. The abdomen is also just a much larger compartment. So you have a lot more space for capsules, a lot more space for biomass of islets. You need a large biomass to really reverse diabetes. And then lastly, the other big variable too is that in the skin, and part of this issue of angiogenesis and diffusibility is that in the skin, you have tens of microliters of interstitial fluid that are squeezing through very dense tissue. We know that when you change blood glucose levels in the blood, it takes almost about 15 to 30 minute delays for those blood glucose changes to be registered on a continuous glucose monitor that you place under the skin. Whereas in the abdomen, there's tens of milliliters. There's actually fluid flux occurring all around those organs. And not only, so that was interesting, actually, in one of our earlier papers, Matt Bohenig published this in, thank you, Lucy, appreciate that comment. Did you try repeated injection? No, we didn't as well. So that's another thing too. We could have actually maybe done a second round within 4 to 7 days, and maybe we would have actually maintained, at least, even if it was a partial drop, because of these viability issues or diffusion issues, or maybe because we're only truly blocking T cells, but not natural killer cells. I agree with you. We might have actually elongated this drop, but given the fact that this was only a partial response and not really truly curing below the cure line, we realized that we would rather modify the particles more fully to address the other immune populations as well. I think that's the first line improvement, and then most definitely repeated injection, unless you go with something that just maintains the signals there. And then you don't have to worry about repeat injections too. I know like Andres Garcia's lab at Georgia Tech has done amazing work with macroscale gels, retaining the signals, like fast ligand as an example, controlling also Treg response for fibrotic response around encapsulated grafts. So
we had him on here too.
Yeah, all excellent questions. I mean, their work is amazing too. Andres saw him a few weeks ago at GRC. So really cool work coming out of their lab. So it's complicated. I think that it's a great question you're asking, and I think that you need to be cognizant of where is going to be the site of therapeutic use, what is the clinical relevancy?
Where's the translational site?
Exactly. Always think about that. If you're at the bench, do not lose sight of that, because the reviewer is going to come back. You're going to write this paper, and someone's going to say to you, why did you do it this way? After all those years of effort you just put in for your PhD, why did you bother doing it this way? And there should be a good reason.
Yeah. And I mean, ultimately it wants to be translatable to clinic too. So that has to always be thought of. I mean, my other question might be a little esoteric, but some groups at Northwell, Feinstein, even Galvani are sort of dipping their toes into, or looking at high frequency electrical stimulation to help reduce inflammation. I can think of Stavros Zanos with his vagal nerve work, but also recently another paper came out in MDPI in 2023 June from Fernandez, Guarno, and Madrid, antifibrotic effects of RF electric currents, radiofrequency therapy. They call it CRET, capacitive resistance electrical transfer therapy. And they're showing in that paper that they see a decrease in expression of ECM proteins, and they, kind of hypothesize that this therapy could be useful for treatment of fibrotic pathologies. So that's kind of interesting. Like, has anyone ever thought, again, in just thinking about combinational therapies, of kind of having that in the backdrop as you bring in these particles?
Yeah. So again, clinical relevancy. Where are you getting your cell source from? Is it IPS or ES derived? Are they? And then you think about only the few human lines that are approved to exist, and the genetic differences that are different from those versus what the recipient. Everybody would love the idea for an off-the-shelf product just working everybody. Yeah. But that's a very tall task order. And additional. So per your point, though, I think I've seen the size of a diameter sphere. We've seen conjugation of biomolecules. We've seen crystallized drugs and very controlled long-term elution release. We've seen modifying the polysaccharide chain backbone of hydrogel alginate. There's many different ways that you can try to approach fibrosis. And I think the holy grail would be to get rid of the need for an encapsulating biomaterial alone, which is why we were trying to move in this direction. What I've seen, believe it or not, Monica, is that some of these phenomena work in different mechanisms and in different biologic pathways. And what's interesting is that they might individually, size or chemistry or drug elution and so on, they tend to work really well in black 6 mice for outwards of anywhere from 4 to 6 months, even upwards of over a year, as I showed you. When we go into monkeys and we go into humans, that rejection wave happens at the similar magnitude, and I would say similar kinetics of initial onset of the immune system, but the rate, there's a lot more fat, and maybe it's just the surface area of the excess tissue, and you get a much larger magnitude of individual numbers of cells. But we see size alone, which works for 4 to 6 months, tend to fail in monkeys with only a matter of only 1 to 2, maybe 3 months. The modified chemistry also was diminished in terms of timeline. What was interesting, though, is that when you combine size with this modified chemistry, or you combine size with the crystals, given the fact that they work by different biologic mechanisms, they actually synergized. And when they were combined together, instead of only lasting 1 to 3 months, where you would even see individual capsules with overgrowth, we actually get prevention of fibrosis outwards of 8 to 12 plus months. And so I'm all for individual different labs, different groups testing from their wheelhouse of what they know. And the reality is that the fibrotic phenomena could be more aggressive in humans clinically, as we've seen. And combination of some of these modalities might be ideal to really nip it in the bud for as long as possible. I also want to just point out that with the graft phenomena we have here, preliminary data, also implicating that addressing foreign body response alone is not, unfortunately, not going to be enough. I wish I had the slides in here from Sherry Stabler's group. Actually, let me see if I have those at the very end. If I kept, no, I did not put them in supplementary slides. The reality is if you have grafts of a different species and they have more immunogenic antigens that they're secreting, you're going to get a larger magnitude and even potentially a different composition of immune response on the outside of your capsule as well. So you have to be cognizant of cell source, how you get it, where you place it. There are a number of competing variables, but I think unfortunately, there's great autoimmune models like EAE for multiple sclerosis where you can hit T cells alone and there's a reasonably good clinical reduction. Type 1 diabetes, even though there's been some antibody depletion of T cells and B cells, and there's definitely effects of both, there's unfortunately a multitude of cells that are coming in to even just get at the either the autoimmune disease tissue or subsequent transplantation grafts. So
Yeah.
it's
No, I think your comments illustrate the need for fully understanding the prodrome of type 1 diabetes and the engagement of the immune system in the different T1D endotypes so that you can really know what it is you're trying to address even before you are designing these implants. So, and that's the work of many right now. So,
yep. And then also getting into the clinic. I mean, given again, so hopefully these humanized mice that we've just created can be a useful tool. One of the things that we're actually testing right now, Monica, is we know that the foreign body response to the biomaterial alone is intact. We don't know if this additional indirect graft response is also intact in this particular version of the humanized mice. We're about to get that data, and we're very excited about that possibility because that would enable us hopefully to have something that might be a little bit more clinically relevant. We'll see.
Yeah, no, Josh, this is very exciting. Your paper together with Jordan Green was just amazing, and the work coming out of your lab as well. Are you hiring for postdocs?
Yeah, so Jordan and I just got bridge funds actually for another project where we're going to hopefully induce tolerance even before type 1 diabetes forms. So there's another aspect of when you intervene. But yeah, I have a few other major grants in submission and review currently, including R01s. And if any of those hit, most definitely. We have one postdoc who's been a godsend. She's amazing. And I'm trying to plateau at the PhD level of lab. We have 8 PhD students, 2 of whom are co-advised, 1 with Jordan. And I'd like to expand a little bit further to complement my skill sets, but also to increase a little bit more senior leadership in the lab. That would be wonderful. We have otherwise an amazing team. They're heavy hitters, and we're lucky in all respects of all the projects that we're working on. Yeah, but I think I'll know more on that question probably within the next 3 to 6 months. But yes, if you feel like you're ready and going to be graduating in the near future, and you like this area of research that we're doing, or looking at the multitude of projects that we focus on, please
start
send me
the conversation. Yeah, and as soon as you've got a shout out for positions, we'll be happy to amplify that. Wonderful. And I just would say thank you again for this amazing talk. Really one of the best talks I've heard, I'd say, in 2023. So we'll shout this out to our audience. We reach an audience of 36,000 globally, and we get a lot of watches and hits on that. So we're excited for everyone to really enjoy this.
I just want to say one thing. I don't believe in hiding negative data, like the instances where we learn something new or where it doesn't work exactly quite right. We always joke there's no journal where you publish negative results. Like someone used a funny nature journal like Nature Crap or something. But the reality is that we learn from those moments too, and those tidbits shared so we can get to hopefully the better place of where we need to be. And it's complicated sometimes. So thank you for hearing.
It's fantastic. We actually heard an excellent talk as we covered IDS Paris by Kizzy Tang, and she really went through a whole host of negative data, very instructive for the field. So nice. Thank you so much, Josh, and we'll be watching what's coming next from your laboratory.
Thanks, Monica. Thanks again for the invite. Take care, everybody.
You too. Bye-bye.